Hello everyone, welcome to my channel Free Learner. In this video, we will discuss plasmid DNA isolation via alkaline lysis method. Plasmid DNA isolation is very important in molecular biology lab. People, those are cloning the gene, they have to propagate the gene in bacteria by transformation reaction. Plasmid isolation is also two types, column-based purification and chemical purification. In column-based purification, which is used in the principle of affinity chromatography for purification, and this is the first one, kit-based purification method, and this is also very expensive method. Then chemical purification. Chemical purification which use the chemical based approach to purify the plasmid, and this is very efficient and cheap to use, and this is used almost all laboratories. Resuspension. This is the first step of purification method. In this method we can say this is also known as the alkaline lysis solution number 1. This contains the glucose, 1 molar glucose, 1 molar 3Cl, 0.5 molar EDTA, NFW. This is the composition of solution number 1 or alkali solution 1. First, we should resuspend the cell by adding the resuspension buffer. It contains the tris EDTA. EDTA chelates the divalent metal, primarily the magnesium and also calcium. Removal of this cation that also destabilize the cell membrane. It also inhibits the DNA. Glucose should also added to maintain the osmotically and present in the buffer to form the bursting in the cell. If you need to prepare the solution, we make sure that this is the R2. This will add in the proper composition. The important part of the alkali solution is that the alkali is done by the solution number 2. This is after proper lysis, it takes out the shell, and solution 2, this is called as the lysis buffer, which contain the NaOH and HDS. The presence of NaOH indicates that the solution is highly alkaline, which gives the name known as the alkaline buffer. Or alkaline solution, what exactly happens? NaOH denature the DNA, cellular DNA become linearized, and also strands are separated. Plasmid DNA is circular and remains topologically constant. So, after putting in the alkaline solution, we mix it very gently, just up and down in the tube, and keep it in incubator, keep it in ice for 5 minutes, not more longer than that. So in this state the DNA can also denature, and also the DNA get denatured because this is the interaction between the DNA base pairs. The hydrogen bonds are broken because of alternation of pH due to its highly alkaline. So the DNA gets denatured in this step. This is the chances of denaturation. So at the time of particular time, we should take out the DNA strand up in this state. Then this is the next step with the neutralization. It is the extremely alkalized environment should be neutralized, and the neutralization is done by the potassium acetate and the acetic acid. We can generally use the acetic acid to neutralize the base of potassium dodecyl sulfate, or PDS, is highly insoluble in water. This is the composition of solution number 3, and for making the 100 ml of solution. After adding this solution, we can see the curdy-like substance or curdy-like the precipitation that so that in the neutralize the solution. So wait for some time, allow the circular DNA plasmid to denature after the centrifugation, and it also precipitate, and in the aqueous should be taken out. Then after that the aqueous phase is taken. Now this is a separation step. Then adding of isopropanol. In this phase, after adding we will add the 1/6 volume of the aqueous phase, adding the isopropanol, so that in this step we will see the pellet. After isopropanol adding, we can see the DNA properly. It makes the pellet. The pellet can be washed by ethanol. So finally, the pellet will be dry in RT and resuspended in the NFW after isolation. So this is the pellet, what actually we can see here. And after the pellet we can see, then we will dry in the room temperature and store in the -20 or -80 for a long year. We can store it. Then now the time is coming to check the purity. This is perfectly good or any contamination at the time of in the isolation. Then for the checking the this is the plasmid is good or bad, we have to check on in the agar gel, and we must get the supercritical band in the thick and other bands are in very specific in size. So then after that we will check in the purity in the nanograph. We can add 1 microliter of DNA sample. So we can find the ratio like that. The 260 by 280 ratio is more than 1.8 and the concentration should be more than 500. So if the ratio is not coming properly, then there should be some contamination at the time of isolation. Thank you.